Biochemistry and Cell Organisation Task Booklet. We're marching on with this. We are now up to nucleic acids. Question 9, nucleic acids. The synthesis of a polypeptide chain, a protein, so synthesis of a protein, or polypeptide chain, involves these two processes, transcription and translation. And we are distinguish, which means tell us the differences between these two processes by placing ticks in the appropriate boxes. So we're going to place some ticks. We're not going to put any crosses in. We're just going to answer the question and put ticks in the appropriate place. Is it just tick one or the other or both? Does it say you may tick either or both? So it's going to have a tick somewhere, but it could be in both. Right, transcription and translation. So we're kind of looking, here's some notes with a picture of what we're looking at there. Transcription, translation, making a copy of the gene from the DNA onto messenger RNA. This goes out of the nucleus into the cytoplasm, where on a ribosome we're going to get translation and separate amino acids are going to get made into a polypeptide or a protein. So that's our process there. So it occurs in the nucleus. Well, here's the nucleus and there's transcription. So we put a tick there. Does translation occur in the nucleus? No, it's out here in the cytoplasm. So nothing there. It involves messenger RNA. There's our messenger RNA in transcription, and that goes out of the nucleus, and here it is being involved in translation as well. So for that, we're going to have both there. Needs RNA polymerase. What does RNA polymerase do? It's an enzyme, ASE, that makes a polymer of RNA. So where are we making RNA? Well, we are making RNA here in transcription because we are making it. Here's a diagram, we're separating our strands of DNA, and here we're making an RNA strand using complementary base pairing. So we're definitely doing that there in transcription. Are we making RNA in translation? No. We're using RNA, but we're not making it. We're making a polypeptide. So that's not going to be a tick there. It involves the formation of peptide bonds. When we are making RNA, are these joined by peptide bonds? No, proteins are. So certainly not in transcription. Translation, what we're making? We're making a protein out of amino acids. These are going to be joined by peptide bonds. So we're going to put a tick there. And occurs in ribosomes. Transcription, anything to do with ribosomes there? No. Here, translation, yes, ribosome. So, yes, it's going to be a tick there. The following letters represent base sequences on a piece of messenger RNA. And we were instructed to write out the base sequence of the original strand of DNA. Okay, helpful picture again. Here's our RNA, messenger RNA there, and here's our original strand of DNA. So we're just using complementary base pairs and realizing that the U would be a T on DNA. So the base sequence would be A goes to T, U goes to A, and G goes to C. So just following that sequence there. So here's the rest: G, A, T, G, A, C, C, A, and G, T, T. Hopefully you're getting pretty good at doing that now, and you're not falling into the traps with the T's and the U's, or forgetting about complementary base pairing. Amino acid sequence now gives us a table. You're not expected to know the names of these amino acids, so you might recognize some of them. or indeed remember any of these codons. What you are expected to remember is that a codon is on messenger RNA. So our codon is on our piece of messenger RNA. So all we're doing is taking this codon here, finding it in the table. So AUG, so that's MET, stands for methionine, but again, you don't need to know that. CUU, and so on. So you're just filling these in from the table. ACU is that one, whichever that one is. And GGU, I should go up to these first. So GGU, can't find it. There it is. Glad I found that. I look pretty stupid. And the end one, CAA, which is. So be careful here that that spelling there and that spelling there are pretty similar. So I've done that, and I'm just going to write in the answer in the place that the answer should be. Okay, and then I'll just put a little line between them. And there's your answer. Be careful, if you've got any spare time at the end of a test or after you've done your homework, work through these again, checking them. It's very easy to make little mistakes that you will spot if you check them. Okay, so always check your work. Over the page, label a drawing of the transfer, the tRNA. Okay. Down here, that's alright, those are our 3 exposed bases of our anticodon. That one's okay. And here, this is where the amino acid would attach. So, amino acid site, amino acid binding site, where the amino acid attaches, something like that. So I'm just going to write amino acid binding site there. That's 11 marks.